Good morning. So my name is Lazartigues. I'm an associate professor of pharmacology here at LSU in New Orleans. And today I will be telling you a little bit of what we're doing. So we have two main projects in my lab. We're interested in cardiovascular disease in general, mostly hypertension and type 2 diabetes at the moment. And we're working with the Renin-Angiotensin system. I will tell you a little bit in a minute about the Renin-Angiotensin system. So I was told to provide some kind of Biosketch information. So here it is. I hope everybody can kind of see this coming slowly. I got my most of my education in France, in the southwest of France. Southwest, southeast. I got bachelor degree in cellular biology and physiology at the University Paul Sabatier in Toulouse. I also got what we call there a maîtrise, which is basically another bachelor degree in physiology, same university. Then I went on to do a master degree in cardiovascular pharmacology at the University Claude Bernard in Lyon. And went back to Toulouse to do a PhD in cardiovascular pharmacology. In '99, after graduating from Toulouse, I went to the University of Iowa and started a postdoc fellowship there with Robin Davison, who's currently at Cornell University. I joined LSU in 2005, just at the beginning of August 2005. So if you have a marker in your head, it's pretty easy to remember, August 2005. Then went back to Iowa for a little bit, and then came back to New Orleans in January 2006, and I've been in New Orleans since. If you want to know a little bit more, I invite you to go check that link. That's below, and you'll be able to know a bit more about the lab, what we have been doing since. All right, so I'll tell you a little bit about hypertension and type 2 diabetes. So cardiovascular disease are the main killer in the United States. They are responsible for about almost 900,000 deaths in 2005. That was about 35% of the deaths recorded in that year. And among cardiovascular disease, high blood pressure or hypertension, death rate is about pretty close to 20%. That represents 74.5 million Americans in 2009. Roughly 1/3 of the population has high blood pressure. And another third is pre-hypertensive. So that means that you get about 2/3 of the population who are either currently hypertensive or will be hypertensive in the coming years. We have to talk about relevance money that represent about $73 billion healthcare cost in 2009. The funny thing with hypertension is that you can live with hypertension for many years without knowing it. And actually, about 30% of the people who have hypertension don't know that they have hypertension. Because until the symptoms get to the point where you get end organ damage and you get a stroke or atherosclerosis, or one of your carotid get clogged, then you don't know that you have hypertension. Once you've been diagnosed with hypertension, you go through a regimen of medication, and it's not uncommon that people get 6 or more different type of medication to be able to control their blood pressure. And it's not always working. About only 1/3 of the people who are taking medication for high blood pressure have actually a controlled blood pressure. There's two types of hypertension, what we call primary hypertension and secondary hypertension. Secondary hypertension is pretty easy to cure because it's secondary to something. So, for example, if you have some kind of tumor of the adrenal gland, you will have enhanced production of adrenaline, and that's going to raise your blood pressure. Well, you remove the tumor from the adrenal, and then the blood pressure disappears. So that's pretty easy to cure. Unfortunately, 90 to 95% of hypertension are called primary hypertension or essential hypertension, and we don't know what's the cause. It's a combination of environmental factors, genetic factors, and other factors that we're not sure we know about yet. I could tell you exactly the same thing about type 2 diabetes. It's 1/3 of the population. You get 1/3 who are pre-diabetic. Hypertension and diabetes, they overlap with each other. Often people who have hypertension have diabetes and vice versa. But mostly because of poor choice of lifestyle, we get to this situation where in the roughly 15 years, the rate of diabetes has doubled in the United States. And if you take the situation of Louisiana here, you see that more than 9% of the population is currently living with diabetes. Essentially, type 2 diabetes, because that's the one that's mostly linked to the lifestyle that we're living in. Okay, so I told you that we're interested in Renin-Angiotensin system. The Renin-Angiotensin system, it's a system that involves different enzyme and peptide, and then you get those enzymes that are going to cleave the peptides and get to the production of angiotensin 2, which is the main player in that system, and that's what's responsible for most of those cardiovascular diseases I told you about. So the way it works is that if you consider the endocrine renin-angiotensin system, the one that your physician will tell you about if you go to see him, you have angiotensinogen that's made by the liver. It's cleaved by renin, which is made by the juxtaglomerular cells of the kidney, and there's a cleavage of angiotensinogen lead to angiotensin 1. Angiotensin 1 is in the circulation, going through the pulmonary circulation, is being cleaved by ACE, angiotensin converting enzyme, mostly in the lungs, and then that will lead to the formation of angiotensin 2. Angiotensin 2 will act on its receptor, mostly the AT1 receptor, angiotensin 2 type 1 receptor, and that will increase sympathetic tone, sodium and ions reabsorption, water retention, increase aldosterone secretion in the adrenals that will then promote the sodium and water retention again. You get stimulation of the AT1 receptor lead to vasoconstriction. In the brain, you have a release of vasopressin or antidiuretic hormone that will also increase the retention of water. And all those factors will contribute to increase blood pressure by feeding back to the kidney and promoting more renin release and so on. So you get some kind of vicious circle that, when it gets out of control, promotes hypertension and type 2 diabetes. So we have different drugs currently used in the clinic, and if you look, those drugs are targeting basically the main element of the renin-angiotensin system. So the oldest one are the ACE inhibitors. They're basically going to block ACE. So they block the conversion of angiotensin 1 into angiotensin 2. You don't get angiotensin 2 anymore. So your pressure should go down. Then people started looking at blocking the receptor that promoting the effect of angiotensin 2. So you can block the AT1 receptor. Those are called ARBs, angiotensin receptor blockers. And more recently, in the last few years, there has been inhibitors of renin. There are will block the activity of the original enzyme in the system. Okay, the drugs are working very well, but that's always not enough to reduce diabetes or to reduce high blood pressure. And the problem with these drugs is that they are relying on a scheme that was designed 20 years ago. So this is the typical scheme. You get this straight cascade going from angiotensinogen to AT1 receptor. Unfortunately, in the last 20 years, fortunately, in the last 20 years, there has been a lot of discovery, but unfortunately, those discoveries have not been translated into the clinic. And so our goal is to go from 1992 scheme to what we know actually about the renin-angiotensin system. Okay, and what we know about the renin-angiotensin system is not everything, all those components I told you about. Well, they're not only in the liver, with angiotensinogen in the liver, ACE in the lungs, and the renin in the kidney. All those components are everywhere in the body, and every single organ actually can have all these components. So for example, if you take the brain, I told you renin is in the kidney. Well, renin is also in the brain and in other parts in the body. And so those systems are called local renin-angiotensin system, and they work together with the endocrine renin-angiotensin system to modulate organ function. So if you take the example of the pancreas, which is not on that picture, you have all those components located on the beta cells, the cells that are making insulin, and these components will modulate the system, the beta cell function, and therefore modulate the secretion of insulin. So a new component of that system has been discovered about 12 years ago now, and it's called ACE2. ACE2 because that was the first homologue of ACE at that time, so just it was easy. They called it ACE2. It's about 48% homologous to the angiotensin converting enzyme I told you about. One thing is it's not sensitive to the ACE inhibitors. And that's good because ACE2 has actually opposite property to the classical ACE. So ACE transforms angiotensin 1 into angiotensin 2. I told you angiotensin 2 is the bad guy in that system, especially when the system gets out of control. ACE2, on the contrary, transforms angiotensin 2 into angiotensin 7. So it transforms a vasoconstrictor into a vasodilator just by removing one amino acid. All the properties of the peptide are changed, and this angiotensin 1,7 binds a receptor called MAS, and this will promote vasodilation. It will improve beta cell function if we're only looking at those two mechanisms. Whoops. So a few years ago, in collaboration with undergrad students like you, we were able to show that ACE2 is present in the brain and acting in several nuclei in the brain, and particularly in nuclei that are responsible for the long-term control of blood pressure. We also show with Cheryl Bindam, who was a grad student in my lab, that ACE2 is ACE2 first increase with the development of type 2 diabetes in a model of type 2 diabetes, which is called the DBDB mouse. And then if we look in the long term, when the diabetes is well established, ACE2 totally shut down. So what we think is happening is that as the disease develops, you get a compensatory mechanism that will increase ACE2. So ACE2 will try to get rid of angiotensin 2. The problem is that compensation is failing in the long term, and then ACE2 crashes, and then the disease gets worse. So our hypothesis is that if we can increase ACE2 activity or expression, we could tilt the balance between vasoconstriction to vasodilation, or from beta cell dysfunction to normal glycemia. So how do we do that? Well, we have developed several tools over the years, and trying to figure out how much time I have. Okay, all right. So we have developed several tools over the years, and basically our toolkit is what we call physiological genomics. It's grouped in that box, and it's basically a combination of classical physiology with molecular biology and gene manipulations. So we rely a lot into transgenic animals. We can make transgenic animals overexpressing ACE2. We also have mice, knockouts for ACE2, and basically we're looking at gain or loss of function with the overexpression or the removal of ACE2. This is just an example of a mouse that we made where the human ACE2 is under the control of a synapsin promoter, and that makes a mouse that overexpresses ACE2 selectively in the brain. As you can see here, the brain is the only tissue among all those tissues expressing ACE2, overexpressing ACE2. These mice, of course, have high ACE2 activity in the brain, and we have been able to show that these mice, once they overexpress ACE2 in the brain, they're resistant to hypertension. I told you we're making a lot of transgenic animals. Sometimes we're making kind of complicated transgenic animals. This is the example of a triple transgenic mouse design that we made, and it actually took us about 2 years to generate those mice here that we call Sarah mice. But we still can make it. And we actually were able to show very nice data with these mice that blood pressure was blunted in that normally hypertensive model. We have done this in the pancreas as well. We have been able, using virus delivery, to overexpress ACE2 in the pancreas, and we went on to show that this manipulation could actually prevent the development of type 2 diabetes in mice that were fed high-fat diet. So what other techniques do we use in my lab? I put a list here, which is not a full list, but that gives you an idea a little bit of the different things we're doing. We're using, I told you, a combination of classic physiology with gene manipulation. We can do microdialysis, so we can basically sample CSF, for example, from the brain of mice and be able to detect peptide and enzyme concentration in those tiny amounts of CSF. We do cell culture, Western blotting, real-time PCR, chip assay, and so on. Let's see. This is my group, and I think I reached the 20 plus minutes that I was allowed to. So if you have any question, let me know. Any question, Baton Rouge?
Hey, how you doing?
I'm all right. I cannot hear you much. Go ahead.
Can you hear me now?
Yes, way better.
All right. You mentioned that ACE2 is used to produce angiotensin 7
Yes.
from 2. What is the side effects of angiotensin 7?
So that's a very nice question. That's a very interesting question, and it's very interesting because so far there's not too many side effects reported for ACE2. And it's to the point where it's actually being used in the clinical trials for the treatment of cancer right now, and it has very good results for the treatment of breast cancer. Basically, what angiotensin is doing is opposing uh, the growth and the vasoconstrictor properties of angiotensin 2. It's called angiotensin 7 because angiotensin 2 is also called angiotensin 1-8. It has 8 aminopeptides. And when these two remove one aminopeptide, you get to heptapeptide with 7 amino acids. But there's no, to answer your question, there's no too many side effects with angiotensin. Actually, I don't know it.
You've got at least one other question here.
Do you know of any groups that are working on the genetics for the hypertension?
I cannot hear you.
The genetics for the hypertension, do you know of any groups that are working on that?
Yes, there's definitely, there are tons of groups working on genetic of hypertension. You get the group of Kurt Silverman in Iowa, who has been working on genetically engineered mice in collaboration with the Renin-Angiotensin System for more than 2 years now. They have developed some of the original mice that we use for with the Sarah. We're doing collaboration with them. There's the group of Anna Dominicak in Scotland who's working on that topic as well. In Milwaukee, there's the College of Medicine who has actually generated congenital rats for many years, where they can transpose. They remove part of the chromosome from one rat, where they think that there are genes located on that chromosome that promote hypertension, and study these rats in the long term. I mean, there's definitely many groups working on the genetic hypertension.
We have another question.
Okay.
Hi. You said when the ACE2 production eventually overworks itself and stops. Is there any understanding of the mechanism through how that system breaks? And if so, any application that you could use to inhibit ACE2 production?
Okay, I barely heard your question, but I think you're basically asking how we can promote ACE2 expression.
No, when ACE2 production is stopped from overworking,
Okay.
is there any understanding of the mechanism behind how that
No,
stops?
and that's a very good question. That's actually my next grant we just put last week. We're trying to figure out actually what are the mechanisms that are responsible for this decrease in ACE2 expression and activity. There's actually a student in this room who's working in this on this topic. We have different hypotheses. We think that could be happening at the gene level, where you could have transcription factor repressing these two activity. That could happen at the mRNA level. You could have increase in various microRNA that will degrade the ACE2 mRNA, and then you don't get production of the protein. Or that you can have different kind of post-translational modification of the protein that would prevent it from being from exerting its activity. But we suspect that we're going to have a lot of those. This prediction in ACE2 is linked to the gene, because what we know is that the expression of ACE2 is reduced. So it's probably happening early, and we already have a few candidates at the gene level. Any other questions?
And any other questions from the other sites? And Shreveport, by the way, you're echoing us. If you can fix your audio. Any questions? Just speak on up.
Let's thank the speaker one more time.
Thank you.
Thank you.